So first of all, what is live cell imaging? So live cell imaging is the study of living cells using time-lapse microscopy. And it is used by scientists to obtain a better understanding of the biological function through the study of cellular dynamics. Live cell imaging involves conciliation between obtaining image quality and maintaining healthy cells. So if you want to improve your image quality, normally you have an increased exposure time, which can be harmful for healthy cells. So you always have to find a balance between those two. And of course, if they say that a picture is worth 1,000 words, then a movie may be worth 1,000,000 words. So that's also something to keep in mind. When you talk about live cell imaging, you really get the whole story and not just certain points of the story. So some history. So the first one who performed live cell imaging was Julius Rees. He was a French researcher, and this was in 1907. And he made movies of the embryonic development because he thought that students would never believe that all cells came from other cells and that organisms were made up of cells that were just coming from one other cell. So this is why he videoed the development of an embryo, and he actually sketched them all out. But his opinion was that drawings and fixed sections are inadequate as they differ from the living in their motionlessness. So that's how the live cell imaging really got interesting in the early 2000s. That's what I need to say. So if you then look at the history, then the first microcinematographic device was becoming commercially available in Europe in 1914. And after this introduction, it went fast. So researchers quickly realized that this technique is a more convenient option to demonstrate cellular behavior. So what Warren Lewis did, he worked at Johns Hopkins University, and he made quite some live cell imaging movies in the early years. So this is in 1934. So what you see here is a monkey embryo that is developing. And you'll see that the embryo is a bit moving, and then the cells start to divide, and they actually undergo proliferation and division, which is amazing to see, which was amazing to see for that time. Here he made a movie about drinking of mouse macrophages. So what you see is that they really have those kind of tentacles, which go out of the cell and then go back in to get all the water back. And in 1936, he also made a movie about the growth of blood vessels. So what you see here is that the sort of lines that you see inside the tissue, they are moving to the right, so they are making a new blood vessel network. And using microcinematography, they were now able to see things that were not visible in any other ways, because the movement of many cells is so slow that the human perception we cannot see that a cell is moving because it's going so slow. So live cell imaging in the present day. So if you now Google live cell imaging, you will get 169 million hits in 0.3 seconds. So in a short amount of time, you get a huge number of hits. If you look now at PubMed, which is a website for scientific papers, and you type in live cell imaging, you'll see that there are 18,214 papers which are about live cell imaging. So there's really a lot of research going on in that field. And also it has exponentially increased since the zeros. So this is the year, here are the 2000s, and you'll see that it just exponentially goes up. So we're going to talk about two methods today for live cell imaging. So on one hand, we have the label-free live cell imaging, where no labels are added, and we have the fluorescent live cell imaging. And this means that a fluorophore needs to be present with the cells to emit light which the camera can observe.